Okay, welcome everybody to this month's instance of the Chalmers AI for Science seminar series. This is a seminar series where we invite young and medium career researchers that are working at the interface of natural sciences and AI and machine learning to present their work here. And today we have the pleasure of welcoming Mohammed AlQuraishi from Columbia University. He's an assistant professor there in the Department for Systems Biology and a member of Columbia's program for mathematical genomics. His research is at the intersection of machine learning, biophysics, and systems biology. AlQuraishi holds an undergraduate degree in biology, computer science, and mathematics. He earned a master's degree in statistics and a PhD in genetics from Stanford University. And he subsequently joined the systems biology department at Harvard Medical School as a departmental fellow. And a fellow of systems pharmacology, where he developed the first end-to-end differentiable model for learning protein structure from data, which was a very highly influential paper a few years back. So we are very excited to have you here today, Mohammed, and please take it away.
Thank you very much. Let me share the screen. All right, I hope you see it again.
Yes.
Great. Yeah, thank you very much for the invitation. It's a pleasure to be here remotely. Love to be there one day in person as well, I hope. Yeah, so let me what I'll do today is tell you, obviously, for the most part, about some of our more recent work on OpenFold and protein structure prediction. But since I noticed that the seminar series is really sort of a kind of a high level focused on AI for science, I thought it makes sense to spend maybe in the beginning, the first 10-15 minutes, just talking a little bit more about AlphaFold 2 in general and sort of why it's such an important development. And then I'll get into some of our own work in terms of rebuilding and retraining AlphaFold from scratch, essentially reproduction of it, and some of the things we've learned, and why we've done this reproduction as well. I'll get into that later on. But for starters, let me just kind of begin with the basics. So the problem, as you may know, that AlphaFold solved in some ways, and at least in some restricted version of the problem, is this question of going from the amino acid sequence of a protein to its three-dimensional structure, right? That's sort of kind of colloquially known as a protein folding problem, although really more accurately here, it's the protein structure prediction problem. So we're trying to essentially predict what's sort of often known as native states, so essentially the lowest energy conformation of this protein sequence, ideally in sort of physiological conditions, although oftentimes the data comes from crystallographic structures, so proteins that crystallize to form a regular lattice, and of course these conditions may not really correspond to physiological conditions, but we pretend like that they do. And this is part of a larger area of problems having to do with molecular modeling, not just modeling the structure of a protein, but things like protein-protein interactions, how small molecules like drugs bind proteins, protein nucleic acid interactions, so other kinds of biomolecules, really a whole slew of sort of questions that are at the heart, I would say, of sort of molecular biology. What are the kind of fundamental molecular interactions that give rise to the complexity we see in cells? But for today's talk, at least, I'll focus on that first bit on protein structure prediction. So this is sort of a sufficiently important problem that people actually develop this competition called CASP that happens every 2 years, where the ability of models to predict structure is sort of assessed in a blind fashion. And what happened was that for much of this competition has been going on for over 20 years, and I would say for much of the 2000s and 2010s, the beginning of that decade, there was a lot of stagnation. So here I'm showing essentially that the pink line is indicating kind of the average accuracy of the prediction methods across time, using what's called RMSD, or root mean square deviation, so just something that's summarizing the deviation between the gram truth and the experimental structure. And generally speaking, when we think about crystallographic structures, all of these are two different concepts, their resolution versus accuracy. But generally speaking, we want things on the order of 2 angstroms, something like that. That's what we're aiming to get to. And so what you can see, obviously, is that in much of the early 2010s, the accuracy was really quite poor. But this changed dramatically the first AlphaFold, and even more so with the second AlphaFold, where very rapidly we went from a situation where we could barely predict anything to a situation where we can predict things with high fidelity. And you can see that on the right, where I think the ground truth is in blue and the predictions are in green, and you can see this sort of very high concordance between AlphaFold 2's predictions and the experimental structure, the crystallographic structure. One thing to note here, which I think is sort of worthwhile just emphasizing, is that the first AlphaFold, in a way, was methodologically not that different from what came before it. I think it introduced a lot of sort of engineering innovations, and those obviously helped, as you can see, but it was not maybe conceptually that different than what was there at the time. And maybe more importantly, even though from a methodological standpoint there was a big leap in accuracy, from the practice of structural biology, from the perspective of biologists using this for everyday applications, this was still sufficiently inaccurate for it to be sort of meaningful, for it to be particularly consequential. AlphaFold 2 is really quite different on both fronts. So first, methodologically, I think it's really introduced a bunch of innovations that were sort of conceptually very novel, I think, and different than what came before it. And that was really kind of interesting, and I think has since kind of percolated through the rest of structural biology, specifically structural bioinformatics. But also from a utility standpoint, the fact that it was so accurate made it actually useful for biologists, right? It sort of changed the practice of structural biology and molecular biology, because people can now, if they have a protein of interest they're trying to study, they can predict the structure, and they can actually gain some insights from that that they couldn't before. So this was really I think a big shift with AlphaFold 2 relative to the entire field in a way. I mean, protein structure prediction has been around for a long time, but it was primarily a field sort of focused onto itself, just focused on how do we improve the methods. It wasn't until AlphaFold 2 where it became kind of a service to the larger biology community, and that I think has been a really interesting shift, seeing it happen over the last year and a half or so. Now, it's worth just mentioning a bit about what it can and cannot do even today. So it's largely focused on single proteins and complexes that can fit sort of, as long as they can fit into a GPU, and there's some limitations in terms of how big they can be. But on that sort of, within that category, the median accuracy is about 2 angstroms, which is quite high. It does struggle a bit with multi-domain proteins. So this is an example where you could see the individual domains. So these are almost like the kind of the pieces are well predicted, but the overall kind of complex, not even complex, it's a single protein, but the arrangement of those domains is not so great, right? This one is sort of open or flopping away. So occasionally it struggles with some of these things, but of course some of these multi-domain proteins may be flexible, and so some of that uncertainty may correspond to flexibility in the protein itself. So it's a little bit hard to actually say whether it's incorrect or simply corresponding to a state that we don't have experimental data for. Where it does struggle quite a bit is with mutations in single sequences. So if you have a protein which has been mutated in some region, AlphaFold 2 doesn't really isn't yet able to predict how that mutation is going to alter the structure. And this in part has to do with the fact that the way AlphaFold makes predictions is not from individual protein sequences, but from a collection of sequences that are evolutionarily related, and it uses that evolutionary sort of relationships to essentially detect kind of patterns of evolution that correspond to patterns of structural proximity in 3D space. I won't go into the details for now, but just suffice it to say that if you only have a single sequence, for example, if you have a protein that's been designed de novo, or if you have some sort of organism that's, say, rapidly evolving like SARS-CoV-2, oftentimes being able to predict structures for those sequences becomes much more difficult. And so that's one area where AlphaFold 2 still doesn't really do very well, and essentially none of the methods really that exist today do very well on this problem. Where it sort of completely fails and can't handle at all are the kind of final category, which is things like ligands, right? So if you have a small molecule, AlphaFold 2 doesn't know anything about small molecules, so you can't even present it in its sort of computational environment. Similarly, if you have like a modified amino acid that's not natural, or if you care about the environmental conditions in which you're making predictions, that's not something that AlphaFold can deal with. So this is one example I often like to use to illustrate this point. This was in fact a prediction done at that CAS competition I mentioned earlier. And what you see here so the prediction is actually quite good. So that's maybe to get that out of the way, right? So this is actually one of the most impressive predictions that AlFold 2 made at the time, where you can see even the kind of what I call the side chains. So these are the kind of the atoms that are not that don't hold the backbone of the protein together. Those are in the right position, which is really quite difficult to do. But why this prediction is interesting is because those side chains are sort of coordinated or held in place by a zinc ion, and like I just said, AlFold doesn't really have any notion of ions or any kind of environmental condition. So it sort of essentially kind of hallucinates, if you may, that there ought to be something there, and sort of predicts the side chains in the right position because it's seen enough crystallographic structures to know to do that. But in general, it's not explicitly right reasoning over this zinc ion, and it doesn't have any way of representing it in the structure. So if you wanted to make a prediction under a different set of environmental conditions, AlFold wouldn't be able to be responsive to those, and that's a current limitation, which I should say sort of interestingly contrasts with kind of historically with more like physics-based approaches like Rosetta, which were able to actually, I mean, maybe they didn't do a very good job of making the prediction, but they were able to kind of model these types of things that AlFold can't yet do. So I mentioned earlier that one of the important aspects of AlFold was that it really was accurate enough to be useful to biologists. But a key component of this utility isn't just accuracy, but also calibrated accuracy. So what I mean by this is the fact that AlFold is able to essentially self-assess and predict the quality of its own predictions, right? And that's what's shown in this figure. So on the y-axis is essentially a measure of true accuracy. It's called LDDT. I'm going to use many different measures here just because there isn't kind of one standard one, but they all kind of are trying to get at the same thing. So that's what the y-axis plots, and each dot is a protein. And on the x-axis is p-LDDT, which is AlFold's prediction of that LDDT quantity, hence the p. And so what you see, so two things, are evident. First, most predictions are in that sort of top right quadrant, so most predictions are correct. That's good. But the key point is that there is a decent correlation between AlFold's accuracy and its own assessment of its accuracy, right? And that's the key thing. So it's actually giving calibrated predictions, and often these are sort of readily displayed by this kind of color code, where blue is sort of high accuracy or high predicted accuracy, and then yellow and orange are lower predicted accuracies. And so this has become really key because for biologists, you can look at structure and then immediately know which parts of the prediction you can trust and which ones you can't. And this, I will say, this is something which I think is, in the context of AI for science more broadly, I think is sort of lacking in general, right? In many machine learning applications where we don't make predictions that are calibrated, which makes it very hard to really kind of use those predictions. And I think kudos to DeepMind in this case, where they really thought through carefully, about including these sort of calibrated predictions. A parallel to this, I think, is something like ChatGPT, for example, right? And this is a kind of an ongoing question about, well, how much can you trust when you ask these systems, these sort of chat systems, a question? And part of the problem is that there isn't any kind of color coding right now of the text as it's being written to depict how confident is the model in terms of different regions of that text. But with proteins we actually have this, which I think is really kind of a nice feature. So this actually allows us to sort of assess in a way how useful AlFold is when say considering the human proteome. So when they initially released the model, they also had a separate paper, DeepMind did, on application of the model to the human proteome, and they did this analysis, which is what I'm showing here. First, let's look at the leftmost slice of the figure. So forget about the x-axis for a moment. So what that slice is showing essentially is a fraction of amino acid residues in the human proteome. So all proteins in humans that have some amount of structural coverage already, so that are known experimentally, and that's where the dashed black curve essentially is showing. So about 18% of the amino acid residues in the human proteome have some structural representative in the protein data bank. And then what's shown here in the kind of the solid colors are predictions made by AlFold at varying levels of PLDT, right? Varying levels of sort of predicted accuracy. So even if you ignore kind of the yellow stuff, which is somewhat unreliable, still the gain in coverage is rather dramatic, right? From something like 18% to maybe something like 64, 65%, right? So a rather large gain. And you can sort of quantify the utility of that gain again by these various colors to say, okay, well, these are high confidence predictions versus low confidence ones. The x-axis, incidentally, what it corresponds to is how much coverage there is of the human proteome if you allow not perfect hits to the structure of interest, but sort of homologous hits. So proteins that look somewhat similar to the protein of interest in terms of how similar are their sequences, but they're not perfectly identical. And so when you loosen that, obviously, you get sort of higher. There's an increase in the structural coverage. But of course, those kinds of excuse me, those kinds of experimental structures are not perfectly identical, and so they will not be as good as a prediction. And in fact, I don't have that slide anymore. But if you were to ask how good is an AlphaFold 2 prediction in terms of these sort of structural homologs, an AlphaFold 2 prediction is essentially something like a 90 to 95%, equivalent to a structural template or a structural homolog that's around 90 to 95% sequence identical. So it's really the gain should be mostly viewed in sort of this regime here, right? That's your gain, not in this regime. So it did sort of double or triple the amount of structural coverage of the human proteome. Okay, so that's it for sort of AlphaFold 2. Now let me kind of pivot, I suppose, and tell you a bit about sort of our own work on OpenFold, which sort of started out essentially as a kind of a reproduction of AlphaFold 2, but now we're obviously pushing it in many new directions, as I'll talk about at the very end. But let me first start just kind of by motivating this. Why do we care? Why do we care? Why do we do this? Why do we sort of spend all this effort reproducing AlphaFold 2? First, I should say, just by way of background, so DeepMind was very good about releasing the source code for AlphaFold 2. However, and I believe them for this, because of the fact that the training code is very entangled into kind of the Google infrastructure system, they did not release the training code for AlphaFold 2. They only released the inference code, right? So they released code to allow people to make predictions using AlphaFold 2, but they didn't release code to allow people to train new versions of AlphaFold 2 or to just simply retrain it, right? And of course, the training procedure is quite intricate, particularly with AlphaFold 2. There's a lot of details, a lot of sort of tricks that they employed, which made it quite tricky. So the context for this, for OpenFold, which was that there was no training code for AlphaFold 2, and then furthermore, there was no independent reproduction of AlphaFold 2, right? There's not been any, there had not been any sort of independent, independently trained model from scratch that matches the accuracy of AlphaFold 2. So that's the context. But beyond that context, there were sort of three main motivations. One was to sort of do full scale retraining to allow new applications, as I'll talk about in a moment. The other was to essentially modularize the system in PyTorch. So I'll get into the code base issue in a moment, so that people can reuse those components in new applications. So it's a mix and match some of the AlphaFold 2 components. And then finally, essentially knowledge acquisition, right? We want to, reproduce DeepMind's results to show that they're reproducible, right? To show that in fact, everything that's in the paper, that's described in the paper, really translates to this model. But also to sort of acquire, new knowledge about how AlphaFold learns to predict protein structure. And I think we've since done so, and I think we've gained a bunch of new insights that weren't there in the initial DeepMind paper, which I think are quite valuable. But let me go through these. The first two I'm going to be very quick, but it's the third one really that's going to be the bulk of the remainder of the talk. So why do we care about retraining and what new applications could there be? So just to give you some sense of what happened. So soon after the first version of AlphaFold 2 was released, a whole bunch of papers about using it to predict protein complexes, right? So not individual proteins, but pairs of proteins or assemblies of proteins that form larger complexes. And essentially all these preprints or papers were basically instigated by these observations made by a couple of people, that you could sort of trick AlphaFold 2 into predicting a complex by simply feeding it two proteins that are linked together by, say, like a flexible glycine linker, by a series of residues that don't have a well-defined structure, generally speaking. So they allow the protein, the two proteins to essentially be, sort of move around, and then you could actually predict that complex, or AlphaFold 2 can predict that complex. And this sort of trick worked really well, almost surprisingly well. And the reason I say that is because AlphaFold 2 was only trained on individual protein monomers, individual proteins, right? Not complexes of proteins. And our experience with machine learning in general, right, is that it's very hard to sort of generalize to out-of-distribution problems, things that look quite dissimilar from what you trained on. But here was an instance where AlphaFold 2 was presented with a very different type of problem that it was used to, but actually managed to generalize really well, right? And even, I mean, it was presented, the way it was done was sort of strict, right? It wasn't meant to accept pairs of proteins, but yet it actually worked, which I think really kind of speaks to its generalizing kind of capacity, which is non-trivial. So very quickly, this kind of trick was applied, at scale, for example, by the Baker Lab to predict the main complexes of yeast. There were other efforts applied to human proteomes, and maybe not quite as successful, but still quite interesting. And so quickly there was kind of almost a little sort of cottage industry of different models that build on AlphaFold 2 to use it for the purpose of protein complex prediction. And this is where sort of the question of retraining comes in. So let me just kind of walk you through this. So this figure is showing different methods and their ability to predict protein complexes. Now, not just individual proteins, but protein complexes. And this is a different score called DOQ. 0 is bad, 1 is perfect. And generally speaking, you want to be maybe on the order of like 0.4 or something. That's at least what you want to be able to get to. So Class Pro was sort of what one could consider kind of the state of the art prior to AlphaFold 2. And this was a method, specifically targeted at predicting protein complex structures. Then the other methods I'm showing you, the bottom 5, so these two and these two, those methods were all based on kind of tricking AlphaFold 2 to make predictions along the lines I just described, right, in terms of having these flexible linkers and so on. So what's remarkable is that just by doing that, you already see these sort of qualitative leaps, right? I mean, leaps that are worth kind of like a decade worth of progress, and this is very, very non-trivial gains. Just by using that kind of trick, which is obviously very encouraging and got us sort of above that 0.4 line that I mentioned earlier. But of course, we still had a long way to go to sort of 1.0, right? Now, what's interesting, and this is the one I left up here. So DeepMind, had the code to retrain AlphaFold, and so in fact, what they did here was just take essentially the exact same architecture, just now retrained it on complexes. So they included protein complexes in addition to just monomers, and asked, how well does that work? And lo and behold, it works really well, right? And in fact, it works significantly better than these approaches based on the hack, right? And this is, I think, maybe the key takeaway here, guys, that there is much utility to retraining AlphaFold, so on a new set of data, as in this case, and that utility is sort of quantified, right, essentially by the gap between maybe the maroon curve here and the cyan bar, right? So that really is kind of I think a very simple demonstration. Without any architectural change, without any additional innovation, you could get a significant gain. And I will say, I don't have a slide on this, but in the most recent cast, much of the focus now is shifted toward complexes, and essentially, all the new progress has been on taking multimer and really not actually even retraining, because the OpenFold is somewhat recent, but simply just additional hacks built on AlphaFold multimer. So it's really it has some staying power in a way, more so than I think people anticipated at the beginning. Okay, so that's one sort of main motivation. The other is this question of modular components, as I talked about. So I won't go into the AlphaFold 2 architecture, but there are many components to it, including this kind of structure module with sort of reasons of a protein structure, sort of various intention mechanisms, particularly in what's called the pair and MSA representation. You may have heard about attention. It's sort of it's a core kind of working cause of most modern machine learning algorithms. But AlphaFold had a particular kind of flavor that incorporates sort of notions of geometry in the attention mechanism. So these are all sort of components that are broadly useful when one is thinking about molecular modeling, right? And as a sort of indication of that, right, if you were to look at much of the algorithmic work that's happened since then in RNA structure prediction, for example, and inverse folding, where you're trying to go from the protein structure to the protein sequence, or in protein design, all these innovations, most of these innovations since then, have utilized some components of AlphaFold 2. So IPA is the structure module I mentioned earlier in AlphaFold 2. The triangular multiple update, this is the triangular attention, the geometric attention I just mentioned. So obviously these are sort of useful components. And one thing we wanted to do is essentially modularize them so that people can use them in a kind of in their own new frameworks without having to invent things from scratch. And one aspect of this was that we essentially built AlQuaishi from scratch in PyTorch, as opposed to JAX, which was the initial framework that the DeepMind used, in part to sort of provide a kind of an orthogonal code base that could be used for for new applications, like I said, but also in a new framework that's maybe a bit more popular and a bit more sort of widely used than JAX. And since then, I should say, I mean, for example, ESMFold, which was a kind of a AlQuaishi 2 using not multiple sequence alignments, but language models. I won't go into the details, but ESMFold from Meta was built using OpenFold. So we're very happy to see that it's already been kind of used by major groups to push the envelope. Okay, so now let me kind of move to really the major piece of the talk, which is the knowledge acquisition and the production part of this work. So first, let me just say that OpenFold works quite well. So we're creating it from scratch, and this is kind of a scatter plot of predictions made on a validation set with OpenFold versus AlQuaishi 2, and they're basically identical. There's very little difference in their performance. And this is an example of just an overlay of the prediction. Ground truth in white. I think blue is AlQuaishi 2, and pink is OpenFold. I should say, of course, because these are different models in the sense that there are different the architecture is identical, but the weights are different. You will see, of course, differences in individual predictions, right? So you see, this is an example where we do better, where OpenFold does better, and this is an example where AlQuaishi 2 does better, right? So there will be some disagreement, and in fact, some of that may be useful in sort of an ensemble setting. But instead of in aggregate and expectation, the models perform identically. Now, we did manage to go and sort of improve some of the inference characteristics. So predictions are actually faster, up to 3 times faster on smaller, smallish proteins, 1,000 residues or less. And these are just sort of optimizations that we did on various fronts. So, for example, we use low memory attention. So this is still a standard attention mechanism. This is not any kind of linearized approximation, but we just simply implement various kind of tricks, implementation tricks that make it faster and more memory efficient, so that we can actually make inferences on over 4,000 residues, I think 4,800 residues on a single GPU, while as I think AlQuaishi 2 struggled at the level of 1,500, 1,600 residues, so a lot less. And this can be applied both to monomers and multimers, right? So it's a large complexes, things like that. And I should say applicable to the published AlQuaishi 2 weights. So even if you don't want to use the OpenFold weights, you could just use the OpenFold code base with the AlQuaishi 2 weights and get similar thing. And we could trade speed for memory, right? So that's nice. So if you sort of willing to wait longer, you could actually infer larger protein complexes. Now, what we had to pay in a way was the code complexity. So our original implementation, for example, in the triangular attention, was something like 10 lines of code, very, very, beautiful, I suppose, very, very kind of cleanly implemented. But the optimized version, the one that has all these sort of advantages, is over 400 lines of code. So it sort of uglified the code at the cost of or the gain of getting better performance. Now, and this is maybe within the weeds. I'm not sure how much people are interested in this, but I'll mention it just in case. On the training side, we've also, I think, made a number of major improvements. So AlQuaishi 2 was trained on TPUs, right, at Google, but most people don't have access to TPUs. So we have it now training on BF16, which is sort of a mixed precision scheme that is more memory efficient, and that's on A100 GPUs. But we also got working on FP16, which actually took us a bit of time and requires some help from NVIDIA. But that enables training on even V100 GPUs. So in most academic labs, they don't have access to the newest GPUs, and being able to sort of train or fine-tune AlphaFold using sort of commodity GPUs, I think, is very valuable, and that's something that's possible with OpenFold. And this should be a training, so you could use kind of standard things like deep speed and PyTorch Lightning to train the model over a large cluster. And we had to even build sort of custom CUDA kernels to be able to do this very memory efficient attention schemes I mentioned. I'll skip this piece. So okay, so now that we sort of that works, it works well, and it has some sort of advantages but it has over AlQuaishi 2. What we were interested in is what can we learn from this process? Does it give us any new insights in terms of how this model actually learns to predict structures? So the first thing that we picked up on, which I think is quite important, is that it exhibits very fast convergence, right? So the model takes quite a bit to train. We trained it for about 80 days on 44 A100 GPUs, so quite a few GPU hours. But you could actually get about 90% of the performance in something like 2 to 3 days, right? And that's what you see here, right? Where the eventual IDDT is all the way over here, but that sort of initial gain is quite rapid. And that's very valuable, right? Because if you are interested in essentially training variants of AlphaFold for new applications, maybe or maybe just even sort of exploring hyperparameter space around the kind of the optimum that AlphaFold, that DeepMind had identified, being able to do this so rapidly without waiting to kind of the fully trained model is quite useful, right? From a GPU hours perspective, and that's something that we've identified. Another thing is that there's this sort of fine-tuning stage in AlphaFold, which is very expensive. It dominates the training time because the amount of sort of the size of the protein fragments that are used to train the model increase, and because the model scales cubically, that increase is really, really expensive. But it seems, in our hand at least, that it seems that what this fine-tuning stage mostly does is resolve physical violations. So the overall accuracy is not really affected. It's just that things like stereo clashes get cleaned up during this fine-tuning stage. So of course that's important. But again, if you're mostly wondering about just how well does the model perform, and you're trying to sort of explore model space, maybe you don't really care about turning on this fine-tuning stage. Another thing we identify, which is interesting, is how the model's self-assessment evolves as the model is training. So let me just kind of orient you here for a moment. I should say that for most of what I'm going to talk about next, I'm only focusing on the first 5,000 steps of training. So essentially, this rapid rise phase. I'm not going to talk so much about the latter phase because this is where most of the action is and most of the interesting kind of dynamics happen. So color coding here corresponds to a phase of training. I think where are we in the training procedure, and each dot essentially corresponds to an assessment of the model of a partially trained AlphaFold 2 model. So what you see, which I think is interesting, is again here I'm plotting this LDT, the ground truth accuracy versus the predicted accuracy, right? So generally speaking, AlphaFold is actually pretty good at self-assessment throughout the training process, right? So it was quite interesting. So I mean, here, for example, in the greenish region or the yellowish regions, it's performing quite poorly, right? It's not predicting things very well, but it's predicting the accuracy of its predictions quite well, right? And that's really important. So even early on, initially starts out sort of overly optimistic, but very quickly sort of it learns to predict things reasonably. It learns to predict accuracy reasonably well. In this intermediate phase, it's somewhat pessimistic, right? So it's actually better than it really is, but then eventually kind of converges to, especially if you wait to the very end, to something which is well calibrated and is not pessimistic or optimistic, but sort of is realistic, I guess, in terms of self-calibration. Another aspect of the kind of training is secondary structure elements. So if you're familiar with proteins, they form these sort of hydrogen bonding networks that are often depicted as helices or sheets visually, but they have to do with kind of the local geometry and so the local hydrogen bonding, like I said. So we can identify these elements and we can ask, which ones are learned first, essentially. And what you see here, interestingly, is that helices, which are sort of more local structural elements, they have to do more with local interactions, they're learned earlier than sheets, which are more distributed across the protein structure, and they could really involve very sort of long range interactions, right? Eventually, both are learned quite well, right? But during this initial rapid rise phase, there is this kind of delta between the two, and we can actually go and kind of deconstruct this into sort of even more fine-grained secondary structural elements. And what we begin to see here, sorry, the color code unfortunately changes between those two figures. But what you see here is that so these two are the ones here, the green and orange map to red and orange. But then you see all these new classes, including ones that are very niche. For example, this polyproline helix, this one, that take a very, very long time to learn, right? So they don't have this sort of rapid rise phase. Eventually, when you this is 5,000, this is 90,000, right? So this is a big gap here that we just sort of skip over. Eventually, these also get learned at least much better than they were at this initial phase, but they take a much longer time, right? So this slightly contradicts what I said earlier about the fine-tuning stage, where I said so it doesn't really matter. You could sort of get away with quick learning. That's true for the most popular, kind of most commonly seen secondary structure elements. But for the more niche ones, it actually does take a long time to be able to improve their performance. And as you may expect, this sort of roughly correlates to the frequency with which you observe these secondary structure elements. So the more frequent ones get learned better earlier, while the less common ones take longer to pick up on. Beyond secondary structure elements, we also observe a sort of a multi-resolution or multi-scale kind of aspect to this training, or to learning rather. So what we did here, what I'm showing you here, is that we took proteins and we sort of cut them up in small pieces. We sharded them, and then we asked how well does AlFold2 in predicting just the small shards as opposed to the whole protein, right? So just fragments of proteins. And what you see here is that, so initially, not surprisingly, the predictions are very bad, and that's GTT as a 0. And eventually, so again, sorry, this is just another quality metric. 1 is good, 0 is bad. Eventually, predictions are good, irrespective of scale. It's almost irrespective of scale. It's still a little bit better at the short fragments and the long fragments, but generally speaking, they're all pretty good, right? But what's interesting, right, is that early on in training, right, there's a much wider spread, including doing this sort of rapid rise phase, where eventually, somewhere during this phase, that kind of spread begins to shrink, and then you have a smaller gap. Although interestingly, even from 5,000 to 90,000, that gap shrinks considerably, right? So the model does seem to essentially kind of learn first more local aspects of structure before it sort of transitions to more global aspects of structure. Not surprisingly, but it's sort of interesting to confirm this. And this is true even if you look at individual secondary structure elements. So if you're asking about helices, for example, and looking here now by helix size, so how long an alpha helix is, you again see that by the end, they're all equally well learned, right? So irrespective of the length of the helix, they're well predicted. But early on in training, there's this kind of separation in terms of accuracy. Similarly for sheets, so this is how sheets are made out of individual what are called strands, and they could have multiple strands. So the size here is a number of strands, and again at the end they're all essentially equally well predicted. But early on there's this sort of separation between narrower and deeper or wider sheets. So that's one aspect. It's sort of multiple spatial scales. But we also saw something which is more like multiple dimensions, or that it sort of it seems to learn to predict structures in a staggered manner, starting with sort of one dimension, and then the second dimension, and then the third dimension. I think just showing you an animation illustrates this well. So here I'm showing predictions as the model is training, right? And so initially you see this sort of tube, then you see this sort of curved sheet, and then it sort of acquires volume to fill in a three-dimensional shape. And then finally, you start seeing those secondary structural elements I referred to earlier get filled in, so that the fine details that the model sort of picks up on to get the right hydrogen bonding networks. And this is true, across all the various structures we looked at. So I'll show you another one. This example where some illustration you see is essentially a sort of tube, which then opens up into a sheet, and then and maybe there's a little bit of curvature in that sheet, and then sort of acquires volume. Just to be clear, so this is the animations here that I'm showing you are quite different than, for example, the animations that were in the initial paper from DeepMind, which were predictions as a model is proceeding through its layers, right? So the DeepMind predictions were how does a fully trained model make a prediction as it's progressing through its layers. And what those animations look like, kind of something like almost what you can imagine like conformational sampling, where it's sort of trying out different conformations and then kind of settles down on something that it thinks is correct. What I just showed you here is quite different, right? Here I'm showing you full predictions, right? So I'm going through all the layers of the model, but of partially trained models, right? Of models that were only trained for a certain number of iterations, and showing what has the model learned up to that point, right? And that's where you see this progression from a point to 1D to 2D to 3D. And it's not, I will say, it's still not, we've had some hypotheses that I'll talk about in a second as to why it's sort of learned in this way, but it's not entirely obvious, to me at least, why it should learn in this way. Again, there are some motivations, but it's still, I think, kind of interesting that it did learn in this way. Coming back to this figure, I just want to kind of point something out. So GDTS is this overall tertiary accuracy. And what you see here is that this, in fact, does rise before helices and sheets, right? There's a gap. And so the animation I just showed you, where the secondary structure elements get filled in at the end, that's kind of corresponding to this rise here in the helices and sheets. But the gradual gain in the GDTS, that's when the 1D, 2D, 3D transition was happening, right? In those earlier phases of the model. So one way to quantify this is to essentially ask, so if you were to look at the 3D structure and you compute the principal components in Cartesian space. So this is not some sort of latent space reduction. This is just taking the 3D structure and asking, what are the principal components? So what are the major axes along which you have the longest and second longest and third longest dimensions of the protein. And then you ask, how do these rise as the model is being trained, right? What you see is, again, consistent with what I said, is that the first principal component, that's kind of the longest dimension, rises first. So there's this big gap here, for example, like where this is at some fraction of the first PC's final extent, but the first dimension hasn't even picked up yet. And particularly here, for example, right, the first PC is almost a third or half the way to its full extent, but the second PC is just beginning to rise, and then a similar phenomenon with the third PC, right? So there's, again, this kind of staggering now quantified in terms of how the various dimensions are actually learned. I think I'll skip this because the animation gives the same idea. But let me just kind of raise a question of, is it just dimensions? So is it just simply learning spatial dimensions in sequence, or can we say something a bit stronger, a bit more precise? And we think we can, which is that it doesn't just learn dimensions in sequence, but it actually learns PCA projections in sequence. So let me explain what you're looking at here, because this is a little bit subtle. So we're looking at individual proteins here, and then we're asking, how does, as the model is learning, how do the atoms of the prediction, right, move along the principal dimensions, the principal PCs I just talked about earlier, right? So if you take the final predicted structure, you compute the main axes along which the longest, second longest, and third longest extents lie, and then you ask, how do the atoms move as the model is being trained and making predictions? And there's some technical things that we had to do here, because the predictions could be in different reference frames, so we actually had to kind of align them all so that they are smoothly moving. But if we do that, and then we ask, where is most of the movement evident, right? And that's what these curves are showing, broken down by the eigenvectors along those three main dimensions, right? The three main axes of length. What you see, right, is that blue rises first, right? So it's first moving in one dimension, then in green rises second, and then third rises last, right? And largely speaking, right, the rise in in the second dimension doesn't necessarily correspond with the rise in the first dimension, or the rise in the third dimension doesn't correspond with at least a commensurate rise in the second and the first dimensions. There are, of course, some gains as well because of that curvature. You're not strictly speaking moving in one dimension at a time, so it's not perfect. And you can see that in particular here, where the red and green somewhat overlap, right? Because there is a lot of curvature in this particular structure. But to a first order approximation, you see this kind of separate rise, right? This kind of separation of these independent dimensions over training time. And that, again, that approach I think is not so trivial, right? Because you can imagine a situation in which you simply, where there's no staggering, where you're simply learning all three dimensions at once. And so all of them would go, even though there are different spatial extents, so they will end up at different plateaus, but they will all sort of move simultaneously to their final endpoint, which is what's shown here in the no staggering case. But in this perfect staggering case, you essentially would learn the first dimension, finish with that, and then saturate, and then you learn the second dimension and finish with that and saturate, and then the third dimension in a staggered manner. And that's a lot more similar to what we're seeing here, right? Where sorry, maybe I wasn't very clear here. So what we're plotting, right, in these different colors is essentially the RMSD between a one-dimensional projection of that protein and the final three-dimensional structure, or a two-dimensional 2D projection of the protein and the final 3D structure, and so on. So what this is quantifying is like how much of the 1D structure has been sort of exhausted, right? And how much of the 2D structure, how much of the 3D structure. And that's why I said this kind of staggering really shows a very, very sort of distinct signal. All right, so that's it for the dimensionality business. Now let me kind of get into, I think, the last major piece, which is generalizability. Right? So how well does the model generalize, right? So the first question we asked is, what if you had a lot less data, right? The PDB has on the order of about 150,000 structures that are non-redundant. What would happen if we had a lot less data to train a model on, and how well would it do, right? So the red is if you retain all data, right? That's the full protein data bank. And then these are fractions of the data, with blue, less than 1% of the full model, right? So this, to me, was actually quite shocking because when it's 0.76%, this is on the order of 1,000 proteins. So really a fairly small data set, right? And the reason why I bring this up is because, particularly for thinking about other applications beyond protein structure, right? Domains for which we don't have 100,000 structures or 100,000 data points, it's a key question as to, can we learn from much smaller data sets? And this is suggesting, right, that even though there's a dip in performance, at least for the green line, right, which is around 1.5%, the dip is fairly small, right? And in particular, I mean, 0.7 is a fair bit worse than 0.8, but it's not dramatically worse. And in fact, if you look at the first AlphaFold, the one that made headlines and so on, that model was worse than everything shown in this picture. I think its LDT was something like 0.6, right? So that's remarkable, right? Because it also speaks to the power of sort of architectural innovation in a model, because in terms of data, there was little data difference between what AlphaFold was trained, what AlphaFold 1 was trained on, what AlphaFold 2 was trained on, right? They use the same data set essentially. But AlphaFold 2 was sort of 100 times, you could argue, sort of more data efficient, right? Because AlphaFold 2 could use 1/100th the data that AlphaFold 1 used and still do better than it, right? So clearly, this is kind of an argument for innovation on an architecture as opposed to just getting more data. That the architecture matters in a sense. So that's one set of experiments. But these kind of splits are completely random, right? Here we just simply took the database and just randomized it. So you could argue that real data sets often are biased in various ways, and we want to really assess how well the model generalizes sort of unseen regions of structure space. So for this purpose, we use a CAT classification, which essentially takes protein structure space and sort of groups it, clusters it based on structural motifs, so that there's kind of a hierarchy where at the top level you essentially group all proteins in basically like 3 main categories, or 4 rather, mainly alpha, mainly beta, or various combinations of alpha and beta. And then there are sort of finer grained classifications. But I'm just to give you a sense of the scale. At the class level, there's basically just 4 categories across all the 100,000 PDB structures. At the architecture level, I think there might be 30, and at the topology level is maybe, I think, a couple of hundred. So a fairly significant coarse graining of that space. So we first started with the topology, and then we said, okay, if we take all of protein data bank, we split it across topologies, and then we ask, how well does it do if we retain only a fraction of those topologies and then test on the rest? So here you begin to see more significant drops, not surprisingly, right? Because again, you are now structurally stratifying the space. But nonetheless, right, even with say 10% of the topologies here, we're getting something which is pretty darn competitive, right? And certainly with even 50%, you're basically seeing almost no loss whatsoever, right? And these are structurally stratified, right? So these are really distinct structural motifs. And this remarkably holds even when you look at architecture, right? So now we're talking about dividing the PDB into like 20 or 30 groups. And at the 5% level, we're training on like 2 groups, right? So it's like 2 out of 30 structural classes, and then testing on the rest. And this model again beats the first AlphaFold, which is pretty amazing. And then finally, and this was kind of shocking to me really, we actually trained on only mainly alpha category proteins. So these are proteins that are not entirely devoid of beta sheets, but are really almost entirely made out of alpha helices, and then tested on beta sheets and vice versa. And again, the model is not terrible. It's in fact actually pretty decent, better than AlphaFold 1, right? And just to kind of show this graphically, right? So this is an example of this. So in orange is the actual ground truth of this protein. This is a largely alpha helical protein. In magenta is a prediction of the alpha trained model, and as you see, it nails it, obviously. But this is the model trained on just a mainly beta class, right? And sure, it hallucinates beta sheets when it shouldn't, but by and large, it's getting the overall, geometry not terribly bad, guys. It's 0.5, sort of roughly of the same topology. And then this is the same here for the beta case, where the beta trained model does quite well, and the alpha trained model misses the detail, doesn't quite get the hydrogen bonding networks correctly. But in terms of where the geometry needs to lie, into where the carbon trace, the alpha trace of the structure is, it actually is generally quite correct, right? Again, the LDT is not terrible. So I mean, to me, I feel okay sort of extolling the virtues of this model, because this is not a model that we build. This is AlphaFold, right? So it's DeepMind's model, but it's really quite impressive. I mean, what they did here, because they, training on something which is highly kind of reduced in a very kind of biased manner, but still allows the model to sort of generalize to aspects of protein structure that it hasn't seen. So I think to me, it suggests that it's really learning fairly general principles about protein structures, not just memorizing things. The one aspect of this, though, which I think is important, is that it does seem to have something of a global versus local bias, right? So here we're using two different metrics, one that's more global, so assessing global fitness, and one that's more assessing local fitness. And what you see is that in the global metric, where the colors here correspond to, again, the fragment length, you see a greater spread. So it seems to have harder time, particularly at this edge, at this point of the edge, it has a harder time learning global aspects of structure when it's trained in this reduced manner. And this particularly shows up with architecture versus local aspects, right? So which is not surprising. So these metrics like architecture and class and topology characterize the global geometry, not the local geometry. So it sort of learns better the global geometry, but has a harder time with the local geometry. Sorry, the opposite. Let me just see. So I think I'll stop. Well, I'll give this one some. I'll stop. I'll just mention that in terms of training, it does also the new OpenFold model is actually more stable. So this is the original procedure that AlQuraishi uses, and you see this kind of instability where the model sometimes gets stuck. But with our sort of new training approach, it actually trains more stably, which is nice. So let me just kind of wrap up by saying, so now we have kind of a full implementation of AlQuraishi 2. We have an inference implementation for the multimer. We're close to finishing the trainable version of the multimer. And again, for training of new model ways and so on and so forth. But now what we're sort of transitioning this into is this sort of consortium, an academic industry consortium, focused on kind of more broadly machine-learned biomolecular modeling, right? So lots of different questions that we're interested in, things like single sequence prediction, integrating structural priors from experimental data, right? Alternate protein conformations, small molecule docking, lots of different applications which we're very excited about that we want to go after. And the way we've sort of done this is sort of set this up as a consortium where industry partners can come in and actually support those various projects so that they can sort of prioritize what we go after. And we think this is an interesting model that, I think DeepMind is amazing, and I think what they're doing is great. But I think it's always good to have sort of multiple entities kind of pushing this frontier. And so the idea here is to kind of provide a complementary approach, even a complementary model for how one does organizationally, how one does this sort of problem. And we'll see if this is successful. I think it remains to be seen. So with that, I'll sort of thank particularly Gustav and Nazim, who have led the work on the OpenFold software, as well as folks who done some of the work on the organizing. And this is my lab. I'm happy to take any questions in the remaining time.
Thanks, Mohammed, for a very interesting talk, sort of digging into the weeds of AlQuraishi and sort of bringing some more clarity about the training dynamics. And it's very, very important work. I think it's really, really interesting. So let's see. There are a few questions from the chat. There's one that seems very apt, since you were mentioning this towards the end of your talk, from Julian, that asks you to give us updates about the single sequence structure prediction, AlphaFold versus RGN2 or anything else. And he also thanks for a good talk.
Yeah, thank you very much. So with respect to single sequence, yeah, we do have a model that's been trained that we should do at least pretty soon, that's based on language models. So it's of the same flavor as ESMFold or OmegaFold, if you've heard of those. Has some advantages and some disadvantages relative to them, but it's a general of that same genre. We'll have that, I think pretty soon. However, I think what is interesting on the single sequence front, in my opinion, is really being able to go after things like mutants, right? So, if there is a mutation, can you predict how that structure gets altered and things like that? And that to me is sort of the real frontier. So actually, our effort now is moving away from language models in a way, and going more toward the kind of direct problem of just from a raw sequence to the structure and making that kind of prediction. I think we're seeing promising results. These models are not yet competitive with something like AlphaFold 2, but I think we are seeing promising results. So that's where sort of our effort is right now. And I don't think we'll have anything super soon, but that's I think we'll have something hopefully in the near future.
Okay. So Mohammed, I have actually a follow-up to this, since you were mentioning this, the idea of predicting mutants. So I guess, for instance, a quantity, not only the structural change, but also something like stability or functional changes would be interesting here. So So how do you see that? How do you see the architecture, the AlphaFold 2 architecture, being compatible with something like a loss of function or gain of function mutation prediction? Do you think that this is something where you need to fundamentally take out the architecture, or you can reuse some elements from AlphaFold 2? Or so what is your take on this?
No, that's a good question. I mean, it's something we're still investigating in a way. I do think the answer is mixed. I think there are certain elements of it that can be used. I mean, and that's been shown by things like ESMFold, right, where they essentially took the architecture as is, stuck a language model, and it sort of, it worked pretty well, right? So it's clearly not completely misshapen for it. But there are certain aspects which I think are not so optimal, right? So I'll give one example. So AlphaFold seems to kind of use the MSA to essentially kind of define the global energy landscape. In the sense that if you have a deep MSA, it often kind of places you in the right place in the structure space. If you don't have a good MSA, it places you in the right space, and it does so very quickly, right? So in the first iteration of the model, while if you don't have a very good kind of starting point, it requires a lot more iterations to eventually kind of find where the local minima is, right? So it does something more like conformational sampling in that case. So it seems to some kind of trade-off between those two sort of paradigms. So for us, I think in the single sequence context, the MSA won't be there, right? So that kind of crutch of being able to say, okay, there's the energy landscape, and this is where you ought to go, and you just need to kind of find your local minima, isn't going to be present. So the way we're thinking about kind of modifying the architecture is essentially to kind of lean much more on that kind of iteration, and the fact that it's sort of essentially doing kind of implicit conformational sampling, push that direction much harder, and put less emphasis on being able to kind of obviously use evolutionary information, any kind of information from the get-go to place it in the global minima. So I think that the overall structural, the overall architectural motif is still correct, but how exactly it's kind of constructed and which aspects of it you emphasize will have to change to do well on single sequences.
All right, yeah, so for those who don't know, MSA are multiple sequence alignments, right? I'm not sure if you mentioned this, but so I mean, some of the, sorry, I'm sort of maybe interrogating you a little bit on this.
No,
please. So I mean, some of the most successful methods for sort of variant predictions, they are MSM based, right? There's Eve, and there are deep sequence, and these other methods that are basically learning latent representations of MSAs and then using some either elbow or some likelihood to estimate a loss function, a gain of function. So do you see that there's a fundamental problem when you're moving away from that sort of information when you're doing variant predictions?
I mean, it's a fundamental problem in the sense that we are losing all the power that gives you, right? That's the problem, right? In the sense that you're giving up on this extremely valuable source of information, right? At least in terms of inference time, right? When the model is making predictions. But the way we're kind of compensating for this is to turn this into training time, right? What I mean by this, right, is that you could simply use predictions from AlphaFold as self-distillation training data, right? You could use all those predictions as data in the kind of single sequence context, right? Because at least for natural proteins, you can sort of assume that these predictions are accurate. So we're still kind of using that wealth of information on, that's coming from coevolution to generate training data potentially, but doing inference, we're giving it up. But I think it's sort of it's a necessary compromise, because otherwise, I mean, we're back stuck to that problem, right? So it feels like we kind of have to make that compromise.
Okay, thanks. So I'll move on to the next question from the chat. So this Afnan is asking about if you could give some examples of how you're planning to retrain OpenFold for other applications, such as predicting alternative conformations or antibodies.
Yeah. So I think what about beyond those? I think that's what the question is asking. So one thing we're interested in, for example, I can tell you, is like protein small molecule interactions, right? So ligands for drug discovery, obviously that's the main application. So that requires more surgery because AlphaFold is very much residue oriented. So it's always thinking about amino acid residues. It's not thinking about atoms, for example. So we have to kind of reformulate it in a way, or at least reformulate aspects of it to work on atoms, but that's doable, right? I mean, I'll give you one concrete example. So the structure module, for instance, is defined and really utilizes the fact that proteins are polymers. So the way it sort of does reasoning over the 3D structure, it creates this kind of equivariant network that's able to, reason over structure in a way that's invariant to translations and rotations, but relies on the fact that because you have a polymer, you could sort of relate the orientation of one residue to the orientation of another residue in this kind of linear manner. That breaks down with small molecules, because you don't have that linearity property anymore. You're not working with polymers, and so you can't rely on this property. So then, one thing we're working on is sort of can we generalize these components to now be able to sort of work equivariantly on things that don't look like polymers, and those are the kind of things that I think are challenging. But we can still use a lot of the machinery that already exists, like the evoformer, for example, which is a very powerful way to sort of reason over any kind of edgetic information, any information made out of edges. And so I think some pieces we'll have to get rid of, some people will have to remodel or improve, but there's still a lot there to make use of.
Okay, that's very exciting to hear that you are somehow, it seems like you're trying to maybe make the sort of the spatial resolution of the AlQuaishi higher, so to say, that you are working on maybe atom-wise representations right. Instead of yes. Residue-based. That would be really cool to see whether it somehow also captures some broader physics, right? I mean, like whether you could actually see on alternative amino acids whether it actually also performs well. Like, I mean, but it's really cool work. We don't have any further questions from the chat. So with this, I would like to thank you, Mohammed, again, and we'll stop the recording.